Hi everyone, I'm Ostojic, a 2020 Regeneron Science Talent Search finalist, and I'm excited today to talk with you about my computational biology and bioinformatics research project. So according to Johns Hopkins University, scientists recalculated the total number of genes in the human genome to be around 43,000, about half of those being protein coding genes, and we're not even close to knowing what the function is of all of those genes. And so what I sought out to do was going through multiple papers. I realized this gap, and there are multiple databases of bioinformatic research out there, but there aren't people really to sift through it. I noticed specifically this one gene, CCDC191, also known as coil domain containing protein coding gene 191, that kept reoccurring in multiple papers, and it just was never addressed. It was always there. Nobody really knew what its function was. There were only three things that we knew about CCDC191. We knew that its exact location on chromosome 3, we knew that it was located in the mitochondria, and we knew its exact coil pymol structure. But other than that, scientists don't know anything else. So what I sought out to do was really determine what the function of this gene was, and that was through a process that I'm going to go through with you right now. So it started off first analyzing the gene expression with using the Cancer Genome Atlas, or TCGA database, with CCDC191 over a variety of cancers. And so in this stage, I was able to link CCDC191 to a couple different cancers: kidney, renal, papillary cell carcinoma, as well as breast invasive cell carcinoma. And so at this step, I knew that there were a couple cancers that were related to CCDC191. Then I wanted to see if there was any patient survival relationship with CCDC191, any correlation there between patient survival and the expression of CCDC191. And I was able to link one correlation to bladder urothelial carcinoma. And then after that, I ran a differential gene expression analysis. And what I was really doing here was I was really trying to determine what other genes change their expression in conjunction with CCDC191 that would maybe tell me if they were related in a pathway in some sort of way, and it would really lead to some similar expression there. And what I found from this was that the data lists, the gene lists, were extremely long, and even though I was able to cut down the number of genes that I was able to look at, it still wasn't really as helpful in my case, and so I decided to use some other pathway databases in order to get a better reading on this. Using the Reactome and KEGG databases, I was able to perform an enrichment analysis in order to determine what pathways or specific parts of pathways had some correlation with CCDC191. So I specifically chose the constitutive signaling by aberrant PI3K pathway, specifically for breast cancer, and I built this heat map, as you can see here. And so what you're really looking for are close coloration similarity from CCDC191 with other rows in this heat map, and what that really would show is that those two genes might express in conjunction with each other. So in this case, I was able to show that CCDC191 actually had three genes that it might express in conjunction with: ERBB4, FOXO4, and FGFR3. After this, I performed a mathematical modeling analysis in order to determine, really, is it an oncogene in the majority of situations? Is it a tumor suppressor gene? What I actually found was that depending upon the gene it was expressed with, and depending upon the situation or pathway, CCDC191 is what is called a double agent. In some situations, it's an oncogene, and in some situations, it's a tumor suppressor. So from this process, I really felt like I had built a process that could be used, it could be replicated for other situations, for other genes that we do not know what their functions are. And I wanted to make part of this process more user-friendly. At the moment, going through the heat map, comparing row by row, it's very tedious, time-consuming, and also not necessarily the most accurate if you're just going by the human eye. And so I really wanted more of a pixel-to-pixel analysis. In order to do this, I created my own AI model, which basically is able to take a heat map and analyze what that target gene is, what is a possible correlated gene, and what definitely is not the target gene or a possible correlated gene. And so I designed a deep learning model with 3 layers, And this model ran with an 83.33% accuracy. And so as you can see here with the ROC graphs that were produced, the model was accurately able to determine the difference between a gene that was not correlated versus a gene that had a potential correlation, but it was not able to determine successfully all the time what was the target gene. So my next step that I've been looking into is really designing a model that will have the user input what that target gene is, so it's not really up to the model to choose, it's going to be more user driven, and I believe that then the accuracy can be brought up even more and fix that ROC curve. So overall, I was really able to specifically study CCDC191 and link it to a specific part of a pathway, an aberrant part of a pathway in patients who have breast cancer, but I also was able to create this overall process with a part automation with AI that can be used in the future to study genes that we don't know what their functions are.